In the second experiment, they selected blue flower with round pollen grains and red flower with long pollen grains. In the previous case, we have already seen that the character of blue flower is dominant over the red flower, whereas the character of round pollen grain is recessive to this one, that is the long pollen grains. So in this experiment, he has selected one dominant, one recessive, other dominant. The genotypic combination will be capital B, a small l, likewise a small b, capital L. Here the gamete will be capital B, a small l, here the gamete will be capital B, a small l. And in the F1 generation, the combination will be capital B, a small b, capital L, a small l. It means in F1 generation, 100% plants will be blue flower with long pollen grains. When it's subjected to test cross, means blue flower with long pollen grain is crossed with red flower with round pollen grains. The genotypic combination is capital B, a small b, capital L, small l, a small b, a small b, a small l, a small l. Here same 4 types of gametes will be formed in the like the previous case. Here only one type of gamete will be formed. When it is subjected to the Punnett square like the previous one, here a small b, a small l, and these are the 4 gametes, capital B, capital L, capital B, a small l, a small b, capital L, a small b, a small l. Now make the genotypic combination capital B, a small b, capital L, a small l, capital B, a small b, a small l, a small l, a small b, a small b, capital L, a small l, a small b, a small b, a small l, a small l. Same result will be here, but in this case 7 plants of this combination, 7 plants of this combination, 1 plants of this combination and 1 plants of this combination. It means he obtained the new test cross ratio, and new test cross ratio is 1:7:7:1. So here he clearly indicated that if two such dominant or recessive allele come from the same parent, they have tendency to remain together, but when they come from different parent, they have tendency to remain separate, and this is called gametic repulsion. So actually this was the experiment of repulsion phase. And here also we got the parental combination 87.5% and recombinant combination 12.5%. And this, the arrangement of genes is like this: on one chromosome, one dominant, another recessive; on another chromosome, one recessive, another dominant. And this type of arrangement is called transarrangement. This is called transarrangement. So actually we have discussed the Bateson and Punnett experiment. We only noticed 2 points, that is the coupling and repulsion. So Bateson and Punnett's work was concluded by Morgan, and Morgan coined the term linkage. The term linkage was coined by Morgan, and Morgan indicated that there are 2 parts of the linkage. One is coupling phase, another one is the repulsion phase.